All right. Welcome everyone to this session. We will have two interesting, someone is a bit noisy right now. I don't know who. Maybe now. We will start with Ahmed Elreedy, who is from Egypt. And this is a country that is not very frequently represented in our topic and field. Ahmed studied civil engineering at Alexandria University in Egypt, and after that sanitary engineering at the same university, and followed by this was a PhD in environmental engineering at the Egypt-Japan University of Science and Technology, where he did not only study in Egypt but also in Japan. His research topic was simultaneous biofuels production and treatment of petrochemical wastewater via anaerobic digestion processes. And after that, he did a postdoctoral research project in Japan again, and then went to Germany, first to the Karlsruhe Institute of Technology, and is now located at the Institute of Technical Microbiology in Hamburg, in Germany. And we are very, very excited to hear about your work now, Ahmed. The floor is yours. Awesome. We can now see that Fousiya is able to share her screen. Maybe you can stop sharing and have Ahmed sharing his screen.
Now, are you able to see the screen?
Yes.
Sorry for interrupting.
That's all right. That's all right. Maybe
There's an issue with the Zoom.
Yeah, maybe you can stop sharing now so Ahmed can start his presentation.
Sure, sure.
Thank you. So Fousiya, did you try to stop sharing the screen, please? Yeah, awesome. Thank you.
Can you see my screen now?
It says it has been started sharing. So far for me, it's only black. Now, here we go. There we go.
Okay. First of all, good morning in the West, good afternoon in Europe, and good evening in the East. So I'm happy being here today, and thanks for the nice introduction. And today I will talk a bit, I will introduce a bit about the topic that I'm working currently on. And then I will try to show some results of my recent project. Then our title today is Innovations in Bio-Electrochemical Systems as a Powerful Waste to Energy Tool. Actually, based on my previous study, I was always focusing on the waste to energy tool, but with the other process, which is the anaerobic digestion or fermentation to produce methane or hydrogen. And currently, since I moved to Germany, I'm focusing on the bioelectrochemical system. So let's start with this. The originality of this process started with the ability of microorganisms that can reduce the insoluble iron via extracellular electron transfer. Then we can say 1910, it was proved that by providing another external electron acceptor like electrode, microorganisms can can transfer electrons to this electrode when we can provide the sufficient potential for the reaction. Then from this year until 1980s, there was no focus on this. Then because of the very low efficiency to produce current, then by introducing the mediators that can facilitate the electron transfer between microorganisms and electrodes, the studies started to be again on board until reaching around 2000, when the Lovely's team isolated the Geobacter sulfur-reducens, which is like a model microorganism in this field, which can produce a very high current efficiency from acetate, especially as a substrate. Then we can say that for this process, we have several configurations, like what we hear about the microbial fuel cell, which is to produce electricity. And you can see here, I can show. The process is to have two chambers separated by a membrane, which is a proton exchange membrane, which can separate first that the anodic chamber is biotic with microorganisms, and the cathodic chamber is abiotic, which is without microorganisms. And then this membrane is just to permit the protons to reach the cathode, to be then reduced to water with oxygen as an electron acceptor. So in the anodic side, microorganisms oxidize the organics and produce carbon dioxide, electrons, and protons. And if we have like a high potential reaction with a high potential, like the oxygen reduction on the cathodic side, then we have this this difference in voltage, which can translate it into a current, which we can use it for electricity. If we change this configuration to be like to stop providing oxygen as a very high, easily electron acceptor, and then make it anaerobic in the cathodic side, then the reaction that can happen is like the reduction of protons to produce hydrogen, which is another source of energy. But for this, we have to provide a power to like facilitate the electron movement from the anode side to the cathode side. And this process is called microbial electrolysis cell. And this, we add the power and we get hydrogen. Then it's we choose between both processes based on what we need. If we need hydrogen, which can be used for specific targets, like for example, the overall aim of my project is to produce hydrogen from a biological process, which we can use the wastewater as substrate, and then this hydrogen plus carbon dioxide are used to produce a biofuel, which is called dimethyl ether, DME, by DME, which is more environmental friendly to be used. And currently there are a lot of developments to use this fuel instead of the conventional fossil fuels. The third type is microbial electrolysis, microbial electrosynthesis, which we use electricity to produce a platform chemicals, which is highly valuable, and we can, it can be used commercially. And then in this case, you can see that the microorganisms is in the cathodic side, not the anodic side, which then can use the protons to produce hydrogen, and another microorganism to consume this hydrogen plus carbon dioxide to produce a highly valuable bio products like a lot of carboxylic acids, acetate, propionate, butyrate, and another more complex compounds like acetylene, the butanediol, PHA, which is polyhydroxyalkanoates, which is used for the bioplastics production. Then this process is is attracting a lot of attention these days because the substrate is mainly carbon dioxide. Then to sum up these processes, we have the anodic chamber, the cathodic chamber, and we can differentiate which process is of our interest based on the available substrate. On the anodic side, like this, we have a lot of substrate can be used, and based on the wastewater that we have, the characteristics of this wastewater, if the main target is the wastewater treatment, but especially when we target the electrosynthesis process, microbial electrosynthesis process, we use in the cathodic side we use carbon dioxide as a substrate, and the anode there is no organic carbon source. Then the potential mechanisms of this extracellular electron transfer, we have three types of extracellular electron transfer to the electrode. It can be direct, using this outer membrane cytochromes from microorganisms, which is based on a direct connection between the microorganism and the electrode. If there is no direct interaction with the electrode, then we will not have this current. And the second one is via nanowires naturally produced by microorganisms. We can say that not all the microorganisms are able to produce these nanowires or bili, electroactive bili. This is the second mechanism. The third mechanism is, which is, we can say the wider one, is to produce the current via shuttles, electron shuttles, which can be naturally produced by another microorganisms or synthetic compounds that we can add to improve the efficiency. These mechanisms mainly depend on the microbes themselves, because for example, the well-known model microorganisms for this process is, as mentioned, Geobacter sulfur-reducens, Geobacter metalli-reducens. They are able to produce current via the first and second mechanisms, direct interaction with the electrode, and they also can produce the nanowires. That's why they are the most efficient microorganisms in this process. And then Chivanella onadensis, it's also well known with its ability to produce, to transfer electrons to the electrodes, but only via shuttles and direct interaction. But this microorganism has a very important problem, that they cannot build a strong biofilm on the electrode, which is challenging, that we need to have a thick and a strong, robust biofilm on the surface of the electrode, then we can have a high efficiency. That's why the Chivanella onadensis still attract a lot of research to increase the efficiency. Another difference between these microorganisms that they are different in the substrate that they can uptake. For example, the Geobacter sulfur reducens can use acetate or hydrogen as electron donors. Geobacter metallic reducens plus these can use propionate as electron donor. For the Chivanella onadensis, it's only working with lactate as substrate. This is also one of the limitations of these processes, but a lot of research is now going to isolate other microorganisms that can transfer electrons to the electrode, which there are a lot of microorganisms that can do it, but still there is no evidence of how they can do it. For these microorganisms, it's already proved, and the pathway of electrons out of the inner membrane to periplasmic, and then from the periplasm to the out of the cell through the outer membrane. Then another topic that is our lab is working on actually is to genetically engineer strains for specific targets. As you can see here, you can control the using cheaper substrate like glucose to produce instead of producing fermentation products by adding the electrode is to produce another target products that can be more valuable. And on the right side, this is already work has been done in our lab, this is Chivanella onadensis, which can use lactate, and by genetically engineering this strain, they replaced the lactic acid to actually in our lab they used to replace the lactate to glucose, which is cheaper substrate, and then we can get more valuable products like acetic acid, but in our case the focus to produce acetylene, which is very high valuable product. Another topic in our lab that they are focusing on is to characterize the biofilm. As I mentioned, the biofilm producing a thick and a strong biofilm on the surface of the electrode is very important, and works are developing systems that can observe how much the thickness and the kinetics of the biofilm formation with time, and together with another laboratory in KIT. And in our lab, they developed this system, which we call it flow cell. We can connect the OCT imaging, its optical coherence topography system that can we can take images to the system while it's working in a continuous mode to see the morphology and the like this the cross section. We can calculate the height of the biofilm. We can calculate the roughness, how much the biofilm is distributing on the surface of the electrode, which is very important to optimize the process, to optimize the flow rate, the temperature, the initial concentration of microorganisms towards the better biofilm formation, and we correlate the biofilm formation with the current density to reach the higher efficiency. And in the upper side, it's the same idea, but we used we call it chips. We manufacture it in the lab. It's very small, and it's easy to make it in triplicate, and you can use it with different conditions, and with the same idea to observe the biofilm with time and with the current with the current production. Then another topic is to use real wastewater, because the difference is we have complex wastewater, and most of the work is like fundamental, that they use a specific substrate. To use a specific substrate is very beneficial if we are targeting the valuable product, but if we are thinking about the wastewater treatment, it's about the applications. And in this project, the substrate of interest is the brewery wastewater, which is this is the beer industry, which is very big in Germany. And as we see, to produce 1 liter of beer, we produce 3 to 5 liter wastewater. And
Ahmed, can you try to share the screen again? There was a little break in connection.
Okay. From where I can continue?
So here is good.
Yeah,
Thank you.
Okay. I was talking about the aim of this project, which is I'm working on, is to use real wastewater as a substrate to the microbial electrolysis cell to produce hydrogen. And then another team in another university is working on the production of DME, the biofuel from hydrogen and carbon dioxide. Another team in another university is working on the economic visibility and environmental benefits from this project, which is interesting to have this kind of collaboration and to give something like a bit compact, that the efficiency, the economic and environmental aspects. Then back to the brewery wastewater, this is the main characteristics. We have acetate propionate, and we have others. So what we know in this substrate is like 70% and 30% is organics, and we don't know what is exactly in the system. And this is the system that I used, which was manufactured in our lab by a previous colleague. Here we can see this is the whole system is like this is the anodic chamber, this is the cathodic chamber, and here is the membrane. Here is the proton exchange membrane, and this is the anode, which is graphite filled, and the cathode is platinum mesh, and this is the reference electrode, which we can apply a potential for the working electrode, which is the anode, and then we collect hydrogen from the cathodic chamber. And our 3 questions in this project is to try to enrich exoelectrogenic bacteria. As I mentioned, we don't know a lot of exoelectrogenic bacteria. Then we want to enrich the ones that can uptake the substrate that we provide. We want to assess the efficiency. We want to observe the biofilm kinetics with time. And this is one of the results that we got. We started with batch mode feeding, which we feed like the wastewater, and then with time the organics the organics are consumed, and this is the current is produced. For this system, the target is to produce hydrogen, but we detect the current production by the potentiostat, which is representative to the produced hydrogen, as a kind of online and more accurate analysis, and then we calculate the equivalent hydrogen produced. Then we started with like batch mode cycles to enrich the microorganisms with time, because we exclude the microorganisms on the planktonic phase, and we leave only the microorganisms that could be grown on the surface of the electrode. This kind of enrichment is like an isolation and enrichment of this bacteria. And then we started with a continuous mode operation, and we can see that we have this stable current, and then with increasing the flow rate, we got a bit higher current density, which is comparable to what we got in the batch mode operation, which is around 400 milliamp per square meter. We calculate the current density based on the surface area of the electrode, because this can be more optimized in the upscaling. From this, we took samples to analyze the microbial diversity and the which microorganism could be on the surface of the electrode. Then we got the anodic community, which is here, and the planktonic community, and we found that it was dominant by these Atsospira species. Atsospira, until now, there is no information how it can transfer electrons to an external electron acceptor. There are some studies that prove that it can do it, but they don't know how to do it, how it can do it. And usually it was found on the cathodic side, which can accept electrons from the cathode, not donate electrons to the anode. Of course, the mechanism should be the same, but still it's not clear. Then we did, for 2 samples, we did metagenomic analysis to get the whole genome of the Atsospira and see what is the potential of this extracellular electron transfer. We searched for the functional genes in this process to see if it has a potential or not. And from a lot of results that produced by this metagenome analysis, we could get that, especially that we found the Pseudomonas aeruginosa in the planktonic phase. This microorganism can produce naturally available mediator with phenazine, and we found that genes that can reduce the phenazine, and then phenazine can be oxidized abiotically on the surface of the electrode. Then until now, we can say this is the hypothesis of behind the electron transfer by Atsospira, but still the metagenome gave a potential. Then if these genes are available or not. Then further analysis like metatranscriptome is now ongoing, and we see if these genes are upregulated on the anode or not. So it's still interesting. Then to increase the efficiency of this process, because we can say that in these processes, we evaluate the efficiency based on the Coulombic efficiency. The Coulombic efficiency is the ratio between the current produced and the organics consumed. So how much carbon was consumed from the wastewater to produce current. of course, the aim is to be closer to 100%, which is mean that all carbons were consumed to produce electricity. So for this Coulombic efficiency, the original community was like 40%, which is not that bad in these kind of systems. But to increase this, we added the geobacter sulfur reducers. As I mentioned before, this is the best biofilm builder microorganism, but still work only with acetate. But it can like build a biofilm which is mixed between the geobacter sulfur reducers plus the Azospira in this case. So we found that the Coulombic efficiency increased significantly to around 80%, and the organic removal even reached more than 90%. And from this figure, we can see that the blue line is the current produced by the Azospira, and the red lines is the current produced by geobacter sulfur reducing. Through these are 2 batch cycles for like 25 days. And we can say after the first cycle, the second cycle, we reached 2 amp per square meter, which is very good value compared to the organic content in this substrate. And how this happened, we can say that not only if you can see this, this is the acetate, which is the main substrate for geobacter sulfur reducers. The acetate with this dotted line is consumed much faster than the original one. Not only this, but the production of acetate at the beginning is also higher and faster, which means that it also increased the fermentation efficiency.
Ahmed,
For example,
thank you very much. We are running out of time. I'm sorry.
Okay, I already
20
finished.
minutes
Sorry,
and you're
I'm
a
sorry.
bit over. I'm sorry, I have to interrupt you here.
Okay, I didn't know actually what exactly the time. It was not mentioned.
I'm sorry if you missed it. I'm sorry.
No, it's
Anyways,
okay.
it
I
was
finished.
very interesting talk and presentation, and I guess we have time for a few questions.
Okay, thank you. I already finished. Thank you.
I'm so sorry, Ahmed.
No. I
Does
just
anyone have
would like
questions?
to acknowledge my team.
Yeah,
and our boss
you
is
go for it,
Johannes
of course.
Fischer. Yeah. Thank you very much
Great.
for listening, and sorry for passing the time.
Oh, good. Oh, good. So, is there anyone who has a question? Mina, please go ahead.
Yeah. Hello. Hi, Ahmed. Thank you very much for this talk. It was super interesting and important. And I have a question that is a bit probably general and not directly connected to Geobacter and the several project that you're working on, but I'm interested in whether this microbial fuel cells could be then used in wastewater treatment to treat refractory pollutants such as antibiotics, chlorinated compounds, something that is pretty hard to get rid of in wastewater treatment. Would that be an option to use MFCs there?
Of course, and it's very interesting to work on these topics, and some people are doing it right now, because as I mentioned, we don't know. The idea is if there is a microorganism can uptake this kind of complex compounds like antibiotics or pharmaceuticals or specific compounds, and can at the same time can transfer electrons externally. So we still don't know. We don't know a lot about all microorganisms that can do it. Even if it's not exactly the microorganism, but the community, if like there is a syntrophic interaction between the microorganisms that can uptake the main pollutant to a simpler one, and then the simpler one is used to produce electricity, or is uptake by another microorganism. So the community itself can be changed in the microbial electrolysis cell or microbial fuel cell. That's why even the anaerobic digestion process, they use anode and cathode to like accelerate the electron transfer between microorganisms. Some accept electrons from the cathode, some donate electrons to the anode. So it's right.
Okay, just in the
interesting
process of producing less harmless ions. Thank you very much.
That sounds very promising. Hans-Peter has another question.
Yeah, I'm wondering
sorry?
because we are studying lake ecosystems, and what we observed in lakes which have an anaerobic hypolimnion, we have GOM respiration, so bacteria can respire organic matter. And I wonder whether something like this is also happening in your biofilm, whether this could impact your efficiency. Can you comment on that?
Yeah, you mean the DOM behavior? Okay, it needs
because you have oxidized, you have oxidized DOM in the oxic water column, and then you have reduced DOM in the anoxic, and quite often it varies between oxic and anoxic. So it's basically like a battery which you can tap, and then it's recharged by reoxidizing everything chemically when you have mixes. So do you have something similar?
Actually, for most of this research, they focus on the, for example, in my project, I focus on the main organic carbon in water, which is like much easier compared to the complex DOM, like humic substances or what you mean. So
but you had phenolic compounds, right? You had phenolic compounds, and they do this as well. So you can oxidize
the concentration. Yeah, sure, but the concentration is very low, and it needs a specific analysis that we can differentiate. And at least for our interest is the other simpler compounds, which is existing in a high concentration. But I got your point. Perhaps another design or another research interest.
Yeah, I was just wondering whether this also plays a role for your biofilm formation, where you have most likely also redox gradients inside.
Yeah, it should.
Yeah. Okay.
But maybe the concentration is still rather low.
Yeah.
Thank you.
All right. Great. Thanks a lot, Ahmed. You see that you
received
a lot of attention from the microbial group here at IGB. That's very awesome. Thank you very much. And I would then switch to Fousiya. Fousiya is a scientific research fellow, a postdoc actually, at the Indian Institute of Technology in Kanpur in India, and she studied zoological sciences at the Calicut University before she went for environmental sciences at Anna University, Chennai, also in India, where she also did her PhD in geology, and will now present her current research at this institute. And Fousiya, the floor is yours. You're welcome.
Thank you so much, ma'am, for this introduction, and good evening to all. So my screen is visible?
Not yet. Maybe it takes a bit of time. There it goes. It comes. There it comes.
Okay.
Awesome.
Thank you so much. So let's begin without wasting much time. Like I have worked on marine carbonates. That's why I have mentioned here as marine calcifiers as timekeepers. Basically, like looking into paleoclimatic reconstruction using marine carbonates or marine calcifiers using corals and marine jam clamshells from tropical islands of Lakshadweep was my research during the PhD, and I'll be showing some interesting results from that isolated area of Indian Ocean, like how these proxies can be used as a paleoclimatic reconstruction in a high resolution monsoon variability and ocean surface temperature reconstruction. So let's begin I'm not able to switch my slides. I don't know why.
Does anyone else have an idea?
Yeah, now, yes. So before getting into the results and what I have carried out, I would just like to show at the end of the presentation what these marine carbonates are and where they form basically, and what are the implications of these marine carbonates as a tool to study the past environmental condition, which is very important, and how the stabilized records in marine carbonates help to understand past ocean surface variations, or you can say conditions in the past. Now, the image that you see in this left corner of my screen is a lagoon from Minicoy Island. There you can see the live corals very nicely, and I hope, I believe that most of the people knows that the corals grows in tropical belts where the sun, where we receive the sunlight throughout the year, and then the water should be crystal clear, and it has to maintain certain degrees of temperature, salinity, and pH and nutrient availability in a way that it has to grow in a prominent environmental condition for its life. So these the image that I have taken, I've shown here is the picture that I have collected from the Minicoy Island, from which is my study area recently. So now, as we all know that this coral requires certain environmental setup for its growth, any change in this surface temperature and salinity will have an impact on the life and its annual bands, and it will retard its growth, because coral ecosystem is very important basically because it hosts 75% of all marine organisms that lives in a area. So which, like most of our lagoon fish and all those vertebrates and other marine organisms that live in a host of this coral ecosystem is very important, because once this coral ecosystem gets any degradations or any degree of destruction, then we are going to lose a big food web from the system. So like the rising temperature is due to the climate change is very important, because now any degrees variations of the temperature will have a impact on the coral's growth, and the rising temperature with the CO2 that is anthropogenic is creating, altering the pH in the ocean system. So by reducing its pH, it becomes more acidic. So what happened is that you see this image, it represents a healthy coral that I have shown in the image in the first slide of my presentation, where in a lagoon the corals grows with its symbiotic zooxanthellae, and the polyps that host the calcification happens because of these processes. So based on that, it grows annually, and it has a very less growth rate basically. And any variation in the temperature will cause this polyps to leave the coral host, and then it will be expelled from the system, and then the coral will completely be bleached out if the expellation of the polyps or zooxanthellae exceeds for few months. So that is how a coral system gets bleached, because of the rising temperature and increasing CO2 in the system. So you can see here that a healthy coral looks like very colorful. It represents as it is believed that coral reef system is the marine rainforest. it's equal to the rainforest in the land, and that's how diverse the systems in the marine system is all about. Now the rising temperature and the CO2 is forcing these systems into more vulnerable and very much threat to the complete diversity by losing its healthy polyps and making it bleached because of the ocean acidification. And if the nutrients of the water, if it's exceeding, there will be algal bloom, and that will close the pores of the corals, which is required for a healthy corals to grow. So all these things have an impact on its life, and which can cause step by step degradation to other systems that ultimately will impact the livelihood of the people who basically depends upon these resources for a lifetime. Now these corals are having density bands, like it has a high density and a low density band that differentiate it from one year to another, like tree rings. You see, like if we know the collection year, let's say the 2013 I've collected a coral, then we can easily count those density bands, and then we can fix the chronology. So this is how the chronology of, or the age model of the corals can be done manually by counting the annual density bands, which forms during different seasons, let's say winter and summer. So now if there is any variation in this oceanic, temperature, surface temperature, sea surface temperature, and or any other variation in the chemistry, that will be easily traced by the isotopic records of this coral records, and the bandwidth that the physical growth can be impacted by any such events. Like for example, if there is any natural calamity, like let's say cyclonic impact or El Nino event, where the sea surface temperature gets enormously warmer than normal conditions. So these variations can be traced in the coral records, in the physical bands, in the annual bands, and as well as in the chemical band, in the chemistry, which is the stable isotopic record. So this is just an example to show how the density bands varies from season to season, and then how we fix the chronology based on the coral records. And the coral, when we studied the coral isotopic records, which is stable isotope of oxygen, let's say C13, and boron isotope to study the pH of the oceanic conditions, that is purely depending upon the stable isotope of coral which grows in an area, is totally depending upon the oceanic condition of that particular region. So let's say the stable isotope of that saline water, that area's water. Okay, now that delta 18 of seawater is basically depending upon the evaporation and precipitation. When there is a precipitation, there will be less delta 18 value, and when there is a high evaporation, there will be enriched values of delta 18. So that variation can be easily studied when we have different seasons. When, let's say, like we have a prominent monsoon season and a winter season, let's say. So the variation due to the monsoonal variability can be impacted in the lagoon, and that can be traced in the corals that grows in such environmental setups. So there are past investigators from the Pacific and from the different part of the world that have been came up with very good high resolution records using this proxy. So now this is how we collect the basically coral core from. We have to choose a good coral boulder, which is less chances for impacting the complete ecosystem, and then finally finding out a exact and right species. We can just take a core from that particular coral, and then the isotopic records can be used to study the past temperature variations and rainfall effects and etc. From these records. So if there is, like you say, if I'm just showing this as an example, like these studies by 1991 to 1999, 2000, they have shown that when there was a temperature anomaly due to ENSO, like let's say this 1989 and 1999, which was a pronounced El Nino year, and this kinds of environment has reduce the annual growth band from the corals, and such annual growths are associated with decrease in uranium calcium ratios in the coral. So because of which the calcification rate will be depleted, and the annual band will be lesser than compared to the normal annual growth. So in this way, this coral records are highly used to study high resolutional data set, let's say from seasonal to annual scale. Basically, if you see the ocean sediment core records, which will have very higher chronology, but then there will be less resolution in the data set. So in order to fill the gap, we have tried collecting a coral core from the Lakshadweep area, which is one of the most prominent coral islands in the Indian Ocean. And like if you see, if this image was from the NOAA's Coral Reef Watch, where you can see that this area has been recorded as the no stress zone for the corals that are growing in the area. But our studies have shown that the corals that growing in this particular island, in this Lakshadweep Island groups, are continuously facing the challenges because of the climate change, and it's more vulnerable to the rising temperature, since the area is continuously rising in its surface temperature since 1960. We have the records that I'll be showing in the coming slides. So now coming into the study area, I have chosen this Lakshadweep area, where we have almost 36 islands, in which 10 islands have been inhabited, and the area is fallen on the monsoonal pathway, which is very important because the area itself has a lag phase in its temperature and its salinity. We have maximum salinity, maximum temperature in the archipelago during March, April, and May, and we have lesser temperature during the August, September, and October, which is our northeast monsoon season. So the area already faced there is a reversal of wind patterns, and because of that we have two which is influenced by two monsoonal system. One is southwest monsoon, and the second one during the northeast monsoon that happens during the September, October, and November. Our JJS is considered to be the southwest monsoon period, where we receive 90% of our rainfall. So that can be believed to have an influence on the lagoon system, and that can be traced in the coral records. This is what the basic assumption for this work was. And then based on that, we have this two objectives, like to study the physical and chemical characteristics of the sea surface using marine carbonates, and to explore their potential as a recorder of past sea surface temperature. Also, now the area is very shallow and very small, so we tried collecting the rainfall for almost 3 years in daily scale, in order to study the characteristic variation in the rain isotope, so to check if there is any impact on the corals that grows in particularly this shallow reef system. So now, if again, this is the transparent and crystal clear water system of the Minicoy Island, where I collected my samples. Now you can see the polyps. Because of the polyps, you can see the color variation in this coral porritis, boulders, that is growing in the shallow lagoon of the Lakshadweep. After collecting the sample, now we have done the coring that I have shown in the initial slides. Now we have seen the annual bands. The image is not that clear because of disturbances, the resolution, because of the resolution. But if you see this image, it's quite reasonable to fix the chronology, though there are bio-erosions because of the other animals' intrusions in the coral system. So now the sample that I've collected was recorded from 1989 to 2013. Now I have removed the two bands because of the, again, due to the, it was not that clear. So from these bands, we have studied the stable isotope records of O18 and C13. Now you see the variations. Delta 18O, the 1989's band has almost lower than -5 per mils 18O oxygen isotopic variation, where the average delta 18O variation is -3.5. And during this particular El Nino event that I have even shown from the literatures, that it was a very prominent year where almost most of the global coral was impacted because of that particular year's El Nino condition. So in that particular year, we have almost 2 per mils variation from than no normal conditions. So which means that area is also impacted by the ENSO. And then what we did was we have correlated the regional surface temperature with that of NINO 3.4 index. And then we found that there was a significantly good correlation of almost R square of 0.5 with that of regional SST. So that shows the influence of ENSO with that particular region. And then that SST variation can be traced from the coral records, where you see the higher temperature, if it exceeds more than 30, then there is a very too much depleted delta ITNO variation, fluctuations in the coral records. So with that, we have tried doing the boron isotope record and then correlate that with the wind speed, NINO 3.4 and the regional SST with delta 13C. So in order to assess this influence of ENSO and other instrumental parameters such as SST and the wind speed and all, by applying the filters into of 2 to 8 years band pass, we have shown that there is a almost like a synchronous variability in almost all the all those parameters. So which means that even if the area is remote, the global impact that is taking place from far away region is being affecting these remote islands and impacting its corals. So this was one of the proxy that I used from coral poritis. And the second one I have worked on clamshells. Now as we all know that gen clams are one of the best proxy again, that goes up to there are literatures that shows like the resolution goes up to 1 daily scales variations we can study using the gen clamshells. Now this area, this is the there are mainly like 9 or 10 pronounced species of Tridacna that is being used to for paleoclimatic studies, where you see that this area, this is my study area, Lakshadweep Islands, where we find the Tridacna maxima and Derasa, squamosa as in major numbers. So based on that, it has a shallow, like this gen clamshells that grows in the shallow coral reefs are also like equally important like corals for the paleoclimatic reconstruction. Now again, this is a field photograph that the maxima species are, I'm showing here alive. So this is how it looks like in nature. This I have taken from the histological atlas of 1992. Now the importance of gen clam as paleoclimatic indicators are this also reflects the ambient sea surface temperature and delta ITNO of the ambient sea water, because one of the advantage of T maxima over coral is that they contain relatively impervious and fine layered shells, so which is less for diagenesis, and then like the resolution of which you can one can go very higher in case of clamshells, and there is no offset, kinetic offset in clamshells that we see in the coral. So that is why we tried clamshells also along with the coral reefs to study how the proxy is, influenced by the global temperatures, and then how it's reflecting its annual scale resolutions. Now, there was a case study done by Obert during 2009 using the same clamshells, which was of 4 years. Now they have shown anomalously depleted delta 18O value during 2002 with a retarded growth in its annual band during a peculiar event that was identified as a cyclonic event that took place almost like within the periphery. And they have noticed the drastic reduction in the growth, and due to the variation in the ambient environment, where the sea surface temperature was reduced by 6 degrees and the sea surface salinity was increased by 0.53 PSU. So in such conditions, they have shown like the extreme events can also impact, like which takes place for only for 5 to 6 days, can also impact its growth and isotopic variations. So this clamshell has the what is that capability or capacity to track the even the extreme events like cyclones, which happens for at least hours or a few days. So even that kind of resolutions can be traced out from the stable isotope records of clamshells, which is not possible in case of corals. So this is the methodology that I have followed to carry out the analysis. Now you have to take a cross sections from the clam, and then from the growth axis we have to these bands, the annual bands in the case of clam is visible, but then we have to finalize it with the cyclicity of the stable isotope records, that is seasonal again, based on the regional climatology. So that is how we fix our our record. And then the stable isotope variations of the clamshell was used to study again the warm surface conditions, where you can see the T maximus delta 18O varies from minus 1.75 per mil to minus 5.53 per mil. And where there was like the 18O of the clamshell has a seasonality, but C13 values of this clamshells doesn't show any variations. It rather has almost similar structures. So which was again noticed in the different species of from different region in the similar way. So now what we have done here is also we did a smooth line curve to see the average variations of delta 18O, where you can see the event 2010, because the sample was so shallow that it ranges only 10 years of life when we collected it. So where we have again seen again during 2010 there was a depleted amount of amplitude of delta 18O variation that was associated with a warm condition, where we have calculated the degree of heat week during that particular year, and then because of which the clam took almost like 1.5 years to bounce back its resiliences. So this is how, like, you know, an area which is already in a experiencing the warming impact of the surface temperature, the sensitive clams and the corals that grow have traced the impacts of the surface temperature. Now we have the seasonality, like in summer we have a depleted value than the winter, but the study area doesn't have a prominent winter, like the temperature variation is just 3 degrees during the summer to winter. So that temperature variation has definitely been traced in the records of the delta 18O of Tridacna maxima, also in the records of the coral porite species in a high resolution manner. Now, in order to see if there is any similarity in the characteristics of delta 18O of clamshell and the corals that grows in the lagoon, we have correlated, or we have tried studying the what is precipitation isotope. So for that purpose, we have collected rain isotope in daily scale since 2015 to 2018, where we have again, like, you know, because of the climate change, the region is impacting extreme weather events since this century. Like the first cyclone that has hit in this particular region was during 2017 December, that was an Oki, which was a severe cyclone. So because of such cyclonic event, like you see the average values of the delta 18 from the precipitation records goes down up to below -10. So in this way, like we have carried out, like we have seen the seasonality for the rain isotope as well as the proxy isotopes that grows in the lagoon, and then we collect the atmospheric samples, that is precipitation samples, to see the seasonality, and then how whether this same characteristics have been observed in the proxy records. And then there was a wonderful result that during the southwest monsoon, it represents the moisture. It represents the source moisture which comes from the southwest monsoon, from the equatorial region, which is enriched in delta 18 variation. So that signature has been observed during the southwest monsoon season, which is during the June, July, August, and September seasons, which is JJAS, and which is prominently driven by equatorial waters, so which is enriched in the delta 18 of precipitation in this area. Now if you see the northeast monsoon season, which is September, October, and November, during this time the source moisture for the region extremely changes from equatorial region to the northeast region of India, which is Bay of Bengal. So the Bay of Bengal itself is depleted in the stable isotopic ratio because of many discharges from the land to the sea and heavy precipitation. So that signature is been observed from the rain isotope records from the Lakshadweep Island, which is very much far away from each other geographically. So these signatures, the extreme event signatures, have been traced from the stable isotope records of precipitation from Lakshadweep area. And then the final conclusion with the results that we have found from the precipitation record is that the proxy records, that is corals, clams, and other paleoclimatic proxies that lives in the lagoon is less influenced by the precipitation, and which is governed mainly because of the oceanographic parameters such as ENSO and regional temperature and the reversal of the current circulations within the area in different seasons. So with that, I'll just show the major findings from my study. The proxy was useful to recognize the anomalous events that have occurred in this region for the last few decades. And as such, a distinct seasonality was observed in the delta 18 records of the clamshells as well as from the coral records. And there was a high amplitude variations in delta boron isotope, with pH shows significant positive correlation that suggests dissolved inorganic carbon fluctuations associated with productivity at Lakshadweep area. The area is highly influenced by upwelling in the monsoon season, and that records have been traced prominently in the coral records. Now the records of delta 18 and C13 of the shells and the coral definitely can be used as a major proxy in order to study the paleoclimatic data set of the region, which is very much important because this area is first of all remote, and then there is developing activities which is taking place in the area that is also causing anthropogenic impacts on the lagoon and the coral reef system, which is very diverse, and the population which is dense, which is almost 75,000 of population is depending upon this lagoon system for their livelihood, is being slowly impacted by the climate change, also by the anthropogenic activities. I also have records of heavy metal pollution of the lagoon using coral skeleton, but because of the time I have not added those into my slides. So these are the major findings of my work. like we have also tried to compare the work from our region with Kavaratti Island, which is again very near by the Minicoy Island, to see the influence of ENSO and how frequently that is being punctuating in the study area. So in that record, we have also seen that in a span of 5 decades, we have almost 10 warmer events based on coral records. So the coral which grows in this region is particularly very important in order to study the ENSO-driven oceanographic upwelling and productivity and oxygen isotope in order to reconstruct the regional sea surface temperature. So I thank my research group. So thank you. I'm like I'm rush because of the time that provided, so I just try to finish it within the time bound. Thank you. And this is also a picture that I've collected underwater in my area. That you can see the polyps, which is looking like little purple or violet in color. These are the healthy coral reef system in the area, which is getting bleached out due to the global temperature rise. So thank you for listening.
Thank you very much. Thanks a lot. It was an awesome and inspiring presentation. So I guess there's some questions. Who wants to start?
Please.
Right now, I can't see. Ahmed, I think, no, Hans-Peter, maybe Hans-Peter, you start.
Yeah, I always have a question, you know.
Yeah,
thanks a lot for this nice talk. I really appreciate it. So I'm wondering, corals are kind of endangered species today, right? Because they face so many environmental stresses. How does it look if you look at older corals? Did you compare? Did you try to go back further in time to see whether there are, is it a constant change, or do we are moving towards now an exponential change?
So like, to identify a coral species itself, it takes like, it's again challenging. Now, we do not know by looking into the surface whether this can be older or this can be shallower. But only by counting the annual rings and annual density bands, we'll be able to fix a chronology. So in which we can get records of hundreds of years in the live sample, as far as I consider my study area. But we do have corals which is more than 3,000 years in the shoreline, but which is dead. So that cannot be used to reconstruct the recent SST, since the aragonite structure would have been like, altered with the exposure. So that we do not know. So in that case, we can have hundreds of years records from the this Lakshadweep area based on the coral growth that I have noticed. Because the annual growth from my study, which is just 7.7 millimeter per year. So imagine like just 13 centimeters long coral records is holding 25 years of record. So in that case, like if I get 1 meters coral core, I can go and talk back about 100 years history. So this is what the common assumption is. And we do have 1 meters coral colonies in the area, but there are a restriction due to annexure species. So we cannot just go and drill it. And there are associated other difficulties to get drilled a good coral core. So these are the limitations also.
I was just wondering, do you see a dramatic shift, let's say, in the past 2 decades?
Yes, sir. We have extreme event changes that I will show, where there was a temperature rise during this 2010. We have a depleted, anomalously depleted value of delta 18O, but now it's in reverse, like, this is -4. And the average delta 18O during throughout the chronology was just -2.5 something. But then when the extreme temperature event took place, the area, the delta 18O amplitude goes back to -5 degrees. So that is kind of signatures that we get. And this is very, high resolutional data set. Almost we have covered like 3 centimeters of a sample. We have collected 192 samples with a resolution of 50 to 150 micrometers distance, which is the best that I could do. So
yeah,
yes, this can be
cool data
used.
set, yeah. Thanks a lot.
Thank you.
Yeah, super impressive. Are there more questions? Right now, I don't see any. You probably impressed everyone so much. Thank you very much for your presentation. To both of you again, Ahmed and Fousiya. Yeah, and we're looking forward. Maybe I can also share my screen. That's not possible right now. Anyways, next Amplifying Voices session will be on April 28th, and we will have talks about phytoplankton on biogeochemistry in the Arabian Sea Shelf, and also about cyclone impacts on shorelines. So kind of at least partly similar topics here and there. I hope to see you all then, and goodbye.
Thank you. Thank you so much.
Thank you all.
Bye-bye.